Okay. Welcome everyone to Georgia Science Junction on Precision Medicine. My name is Patrick Ryan. I am the Director of Engagement at Science for Georgia. Science for Georgia has been a nonprofit since 2018. We came out of the March for Science knowing that there was a need for more communication of science between scientists and the public, scientists and investors, and scientists and the legislators. So our missions are improving communication between scientists and the public. We increase public engagement with science through events like this, and then get that science responsibly used in public policy. We do this through a lot of different events and organizations. We do science communication training for scientists and college students and businesses. We have outreach events like at the Atlanta Science Festival. This is the Science of Ice Cream. We do roundtables on workforce development and other things, sometimes even getting the lieutenant governor. We host conferences about environmental justice and climate protection. This is one of our other roundtables. We've worked with literacy campaigns. We worked with and got March named Georgia Reading Month. And we've worked with the science community to somewhat oppose the Okefenokee mining operation because it is a bit dangerous for the Okefenokee. We do a lot of our work with volunteers and other groups, so if you'd like to know more about our events, you can join our Catalyzer network. We have done a rebrand from Atlanta Science Tavern to Georgia Science Junction. We're doing this to do more events around other parts of Atlanta, other parts of Georgia. Next year, we'll be going to Augusta, we'll be going to Athens, we'll be doing a lot of fun stuff. You'll also be able to see a lot of these hopefully on our streaming, which those of you on stream right now know of. We're on YouTube. We have our new events calendar so you don't miss any of our events. We do a lot of outreach and in South DeKalb and some other areas around Atlanta. Got it? Okay. And if you'd like to help us out, we do take donations. We are a nonprofit. We do this through your support. So thank you for Mutation for hosting. I know it was a little bit trying to get set up, but we're good now. And the speakeasy is actually quite nice. So hopefully we'll be able to be back here again. All right, and now Dr. Robyn Schlicher will be giving her talk on bringing precision to medicine. She is an expert in many spots of this field. I have not enough hands, so give me 1 second. Okay. So Robyn Schlicher brings over 20 years of experience between precision medicine, diagnostics, and entrepreneurial leadership. She is a recognized global expert in advanced analytical techniques and consults with many up-and-coming biotech companies. In addition to working at well-recognized global organizations like Abbott Labs and Leica. Her publication on drug delivery by targeted cavitation, has been widely cited and highly highlighted worldwide. She earned her doctorate in bioengineering from Georgia Tech and Emory University, receiving National Institute of Health and Medtronic fellowships. And now, without further ado, here's Robyn.
I think you're going to have to click for me, or I'm going to, okay. You have to give me a second to understand technology, because I do a lot of technology, but, so, whoop. All right. I think I have this down. So thank you for the introduction, Patrick, and it's lovely to see everybody. I'm glad that I like speaking to smaller audiences right now in my life. As Patrick said, I've done this for a very long time and have a lot of strong ties to Georgia. So I joke about how I predate biotech as an industry, but I actually do. I started in chemical engineering and then helped Georgia Tech actually build the bioengineering department and program and have one of the first doctorates from it in the laboratory of Dr. Mark Prosnitz. So I have a lot of ties here, and I'm happy to support bringing biotech further into Georgia. I was also a person who part of the reason I had to leave Georgia was because there wasn't a good corridor of biotech here at the time that I was working in it. And I ended up going to San Francisco, and that was where I got deeper into the diagnostic space. But we'll talk about that a little bit. We're going to kind of go on a journey here, talking about what precision medicine actually is, what do we mean by it, and the evolution of it from ancient times, actually. So, whenever you hear about precision medicine, what does that really mean? I mean, haven't doctors, to date, at least in modern medicine, tried to make sure that you get the right treatment for your disease? Well, there's different ways of thinking about it. There was an evolution of it from what's called evidence-based medicine, which was kind of based upon population determinants. This is what it looks like on symptoms, and this is how we're going to try to treat people who present this way. And that helps a certain portion of people, but those of us who have experienced rare diseases, unusual diseases, that actually wasn't the best way to go about it. And then that started moving into what's called precision medicine, where technology tools started to become more advanced and we could actually guide it. Usually people think about precision medicine in terms of genomics, but there's actually a lot of different ways you can think about how to make it more precise for you. So we'll talk about that. And then there's this thing now where we talk about personalizing it. How does the medicine really help you as a person? And then one of my last companies like to say, functionalize, personalize, precision medicine, because we could throw as many jargony words in there as possible to say that we're trying to make it about you, because part of it is in our culture now, we want to make sure things are really about us. But in medicine, we really do want to make sure that the medicine is best for us. And so that's one of the things that I've worked on for about 2 decades. And we'll talk about that a little bit more. A lot of people hear this term biomarkers, and again, a lot of the times right now we talk about it in terms of genetics. Biomarkers are actually far more than genetics, and biomarkers have actually been used throughout history. Respiratory rates, the smell and taste of your urine, all of these things actually are indicative to portions of disease patterns that you can think of as a biomarker. And then what has this meant over time, right? So when did we start talking about precise medicine? I looked through some history, and actually it goes all the way back into the BC times. If you go to the Odyssey written by Homer, and I actually had the privilege of having a classics education at a private school before I got to Georgia Tech. So I actually read all of this when I was younger. But it talks about Telemachus, the son of Odysseus, visits Menelaus and Helen to hear news of his father. And Helen puts a drug into the wine to ease his grief and anger. So she tried to use a specified type of medicine, the wine, to help him ease his condition. And so that's the first time that you feel a mention of using something in a specific way of treatment for a person to improve their status of health. And then why this is important is even to the Egyptians, which were sort of the originators of making medicine and medicinal improvements when you think of it through ancient times, they looked to the Greeks. And I thought that was kind of a cute fact that because Greeks were supposed to be the descendants of the god that, or the doctor to the gods. So the Egyptians were looking to, or the Greeks were looking to the Egyptians. The Egyptians and the Greeks had this tie together in this time, and the Greeks actually followed their lead. And that's part of the reason that you hear about it even in the original sorts of documents, this long travail of Homer. But when did it really become precise? So in Egyptian medicine, they actually started to break it down into categories of medical practice. So how do we make this better? Like a doctor or a physician was looking at how we practice a specific type of treatment in Egypt. And there was a specialization for a disease per body part. So it's starting to be like, let's fragment this out and stratify how we can do different ways to treat the body of an individual. And then it goes into what's called Greek medicine. It's more of the classical period. And this is actually considered the first use of real personalized medicine, where they really took it to the body's location and tried to use understanding where it was located in the body and doing specific sorts of study of that body and being specialized as physicians to get a deeper understanding of each type of illness. And so that they saw was getting better therapeutic outcomes. So instead of somebody who just sort of looked at you and tried to understand what humors, they looked specifically at how feet were different among people and treatments that actually helped your foot work better and how your heart was better if you looked at this whole population. So that's kind of considered the first time that we're starting to personalize treatment to at least the individual body component, not necessarily to the individual person yet. And then we get to the most famous doctor of all, right? Any physician, the Hippocratic Oath. So we go to Hippocrates, and he looks at this and he says, okay, this is really cool, but you're still saying a lot of things that are superstitious. There's a lot of this pseudoscience. We're still trying to understand like what humors do to the body. We're still trying to understand how God's affected. So Hippocrates said, we want to go more to a rational outcome and think about this as how diseases can be treated from their origin, and that the treatment carried out should be opposite to the cause of the disease. So, if you're constantly being annoyed by the person next to you, maybe you need to get away from them kind of idea, right? If you're being constantly exposed to something that you're potentially allergic to, maybe you should stop taking that. So it's kind of this first input to what evidence should be used to make your health better. And then with this created more of a focused and personalized approach because, again, what is the thing that's adding insult to an individual's body that maybe we should stop exposing them to? So it started to become more personalized through Hippocrates' work. And that's part of the reason that we glorify or I guess look to him as the originator of what we call modern medicine. He tried to individualize that medicine, and he tried to approach it from an ethical and moral standpoint. As most of you know throughout history, there was a lot of crossovers between doctors and not such good players in that space, right? So he was trying to create an ethical framework under which we would make sure that we do no harm. That's the most famous phrase that people think of from Hippocrates. And that's actually really important these days as we create more technology. So I'm literally on what they like to say bleeding edge of technology in San Francisco. I do not like that term, but a lot of the questions that we ask ourselves, are we going to do no harm with this? Is this really something that needs to be done just because we can do it? During my time here in Georgia, I was actually a stem cell scientist. I was working on regenerative medicine products. And just to talk about some of the politics of even that, you guys are familiar with different levels potentially of stem cells, embryonic versus body cells, adult stem cells. And there was a lot of concern in that time. So it was around the Bush administration to the Obama administration. And Georgia in particular was not really fond of the thought of using stem cells as a research tool, particularly for embryonic. And so many of us couldn't actually continue a lot of that research. That's part of the reason I ended up in California, although I did switch then to diagnostics. So, going from how do you use body cells, what's the ethics of that, and should we use those cells, becomes the next questions of should we continue to use the technologies that have led to gene editing? So should we redesign your baby? These are things that you have to think about pretty often in my line of work. So this became the precursor to kind of the thought process behind genomic medicine. There are individual characteristics of you that are potentially treatable or indicative of your health state and how we can make that better. So there were no genomics at this time, but he was talking about phenotypical potential outcomes as that leads to the idea that there's something about you that makes you special, that makes you treatable in a new way. So, what is modern medicine? I've read a lot of history of medicine books, and one of the most interesting things that I ever read was actually about a serial killer. So I like to tell this story. In the late and early 1800s, medical school was still kind of a free-for-all. It was like, can you get enough money from your parents to get some training? Often it was based in homeopathy, and if you're familiar with homeopathy, even today in its usages, it's fairly questionable, but it was particularly questionable back in the 1800s. You're adding more of the same to do no harm, those kinds of ideas. And it wasn't stratified it wasn't solidified into a program that was really necessarily based in pure science. So often people would prefer to go see the barber dentist, right? So you get your hair cut, you get your teeth pulled, you get your surgeries done, because they had practical hands-on training. And people who even went to what were considered top medical schools in the United States had a lot of theoretical training and often gave you things that had arsenic in it. So, you would go to a barber if you were having, obstetrics kind of questions, you saw your midwife. So even though medical care was available in the United States, it was still kind of shaky. Now, then they started to change that paradigm and actually make, like Johns Hopkins made a really good school. And why this relates back to a serial killer is that in Chicago, Harvey Mudd actually trained as a homeopathic medical practitioner, and was part of his killing field need was to, vivisect his victims, right? So this was all part of this medical history book about how he was trained in medicine. So I thought that was an interesting segue about the history of how training happened in the United States. So about the 1870s, we started taking more of a scientific approach, and we started to create medical programs that were actually based in treatment outcomes, and again, bringing in Hippocratic Oath and saying we need to use ethical practices for our patients. Then we jump up to about the 1950s because we're starting to create more science, and this becomes the evidence-based medicine I mentioned before. We're looking at evidence on outcomes, and this is where you get a lot of these general practitioner type of questions. What are your symptoms? How do I match that to other symptoms of known patients? And how do I give you treatments that will potentially alleviate those symptoms? But the problems with that, again, become often evidence medicine was based in data about men. And it was often based in generalized population information that isn't necessarily personalized enough for those of us with rare and challenging diseases. So then around the 1990s, so we get into my era. So that was around the time I came to Georgia Tech, and we started actually using the terms about prediction of drug responses and understanding things as personalized medicine. And this was kind of a new thought process in general. We're like, well, we can use technology and tools, and we can actually understand things about you at the genetic level, and other things about you to help make drugs that are actually tuned to these genetics and make things more efficacious on the end, save your life, improve your outcomes. And why did this come about? Well, in part because we started thinking more about how biomarkers work. So biomarkers are just objective measurements about things of your body. And again, a lot of people think I mean genetics. I actually thought that for a while. I was like, oh, a biomarker is a genetic marker outcome. Well, actually, it can be almost anything. I work with a company right now that actually looks at, if you think about how cancer is formed in the body, it reaches out with angiogenetic factors to create vascularization to help feed the tumor. And you can look at the torsion and twistiness and amount of those vessels, and that actually is a biomarker that tells you about whether or not that particular tumor is amenable to cell therapies. This is a whole new line of inquiry. I would never have thought that you could look at vascularization as a biomarker. So, almost anything can become a biomarker if you can measure it enough and get enough instances of it to create the data pools that tell you about how to predict an outcome from it, right? And so this is very useful for, again, diagnostics. Are you ill or not based upon a particular profile of your heart rate changing, your genetics that tell you may or may not have Huntington's, those kinds of things. And again, what the really, I think, exciting utility of these technologies, and where I've really worked the last few years, is how do you match treatments to people based upon these particular characteristics of their body. So, but why did we start thinking this way around the 1990s? Well, because this thing called the Human Genome Project happened. It actually changed my life. So when I was a kid, I was thinking about how I wanted to do something interesting with new types of technology. And this was something that was kind of what I was hearing about even before the 90s. There were these things about how you could maybe use genes, think about genetics. And then it became this massive United States project in 1990. And we created a lot of new technologies around it, like being able to use faster sequencing actually came out of that, and to be able to try to understand more about the human body from the genes. Now, this is a way to try to understand things that we were doing to try to tailor disease prevention. Again, are you precursor genetically going to possibly have Huntington's? Like, we can look at that. And then try to understand better ways to do treatment over time. And it was trying to take into account differences in your genes, environments, and your lifestyles. But this is actually kind of a question right now. Did we get everything that we promised out of the Human Genome Project? And the answer is actually no. Like, when I was younger, people were saying, oh, this is going to solve all medicine. We're going to understand your genes. We're going to do the Genome Project. But there's a little piece missing. Even if you can look at everybody's genotype, it doesn't necessarily mean that you understand what it has to do with anything. We had to get enough data, and we still haven't had enough data to understand what each of those genes actually relate to in terms of not just disease, health, lifestyle, longevity, and treatment. So it was really interesting because people were saying, this is going to end all things in medicine. We're going to have the Human Genome Project. We're going to know all your genes. And we're going to make designer babies, and we're going to make these cyborg humans. And 30 years later, we've done a little bit of that, but not really. So I do think it's still an important project, but it's not what people thought it was. And there's a lot of that goes on in biotech. There's a lot of this great big hype. Like, the more recent one was that CRISPR, and it was going to come in, and everybody was going to have the ability to have their genes changed. A little bit. but it's really not. It takes a lot longer than people think when you first hear about the Star Trekness of it. And I think that's something that the population needs to understand a little better. So, but what did this really do for us? It helped us create a whole bunch of really interesting technologies. So to get to this place, we had to use all of these different types of tools that were being built in real time. And you guys have probably all heard about many of these things. PCR, like understanding more about DNA sequencing, genomic outcomes. Like these are all very important technologies that we use every day, actually, now. Not necessarily in the origin of the Human Genome Project, but every day to understand more about our characteristics of our body. So in my mind, it was actually a successful thing because it created technologies. And one of the interesting things I think that we really need to understand is we're all basically the same. And I think we forget that, and I'm not going to go on a big segue about politics and religion, but we're all 99% the same. The interesting flavors in us is actually a pretty small part of our genomics, but that's really critical in understanding how to treat your disease. So that's where a lot of the dwell time is what is that thing that makes us a little bit different? The SNPs, the variable parts of our polymorphisms of our DNA. Like, what does that actually express in the proteins? How are we able to use that information to understand why my version of a disease is potentially different than my friend Sam over there? And so the whole goal of this project was to get a better understanding of an individual's genetic makeup. But I think we got a lot more from that, actually. We got a lot of interesting technologies and a lot of interesting tools and a new paradigm for how to think about things. And I built my career on it. We'll talk a little bit more about that. Now, the hope was that we would be able to very easily and rapidly predict outcomes using your genetics or predict that you'd have a specific disease or match you. And we do that. It's just not as much or as easy as people thought it was going to be. So how do we get back to Georgia? And we'll talk a little bit about my life because I think that it's kind of interesting. So my career, again, started at this time. And I was very fortunate to be in a place to have access to some of the top minds in personalized medicine as we were creating personalized medicine. So when people are like, wow, your career is really interesting, I happened to be in the right place at the right time, but it was also really frustrating because I'm 19 years old in this small private school in Oklahoma saying, well, I can take chemical engineering classes, but I also need to take molecular biology classes, and I can go build bioreactors at this thing, at this national lab, and everybody's like, what is this woman doing? Well, I was building bioengineering for myself, basically. And so that was part of the reason that I got into Georgia Tech. Georgia Tech had it on the roadmap to create the bioengineering program. I came as a chemical engineering major and worked with Mark Prausnitz, and he was out of Bob Langer's lab, Robert Langer, who's probably the only bioengineer that the average population has ever heard his name. If you're familiar with Moderna, he was one of the founders of that. So at that time in MIT, Robert Langer, Mark Prausnitz, Samir Mitragochi, a lot of very big names, we're building out these tools to create targeted drug delivery and actually the things that later became mRNA vaccine. Like there's a massive body of work that they've done because they looked at that and they said, well, there's got to be ways that we can look at things past, at least in Mark's lab when I was there, let's look past standard pills, injections, and we could use that to alleviate the side effects and improve patient outcomes by targeting drugs and therapeutics specifically to the region of interest. So we were building microneedles and others, and I'll talk about him a little bit more in a minute. But why that was important at that time was it was a thought about using mechanical means to take drugs directly to the site of action. We weren't quite at the genetic level yet in this lab, but we were trying to get there. Now, also at Georgia Tech, I was working with a very well-known scientist named Robert Nerum. He was an expert in regenerative medicine and one of the key pioneers there as well. And so that way, we were looking at technologies to create organs for your body, to do organ replacement, to actually understand more about oncogenes. So the same genes that you turn on to make those cells create a new organ could also be potentially cancerous genes. So we were working together in some different ways to understand more about how to use those technologies. Marie Chetta, I mentioned that I was partly a stem cell biologist. She's a well-known stem cell biologist. And so she was looking at ways to use cells with specific characteristics to improve health, and that later became what we think of as cell therapy. So if you think about some of these cancer treatments, immuno-oncology, where we're using specific types of cells to alleviate cancer, those are cell therapy methods. And so she was on the cutting edge of that by using different cells of the stemness to understand how you can improve outcomes using stem cells. And then I was learning molecular biology with a professor named Harish Radhakrishna, who later actually went to Coca-Cola to focus in on I used to tease him that it was how to make your taste buds more addicted to Coke products. But he worked in the statin line, which is really understanding how to improve those outcomes from drugs by using molecular biology. So if you look at all this, this kind of gave me a background that I really understood a lot about cell biology, but I also understood a lot about how you could use different techniques, thought processes, and the work of these really great key leaders in Georgia to make things different and interesting in my chosen field later of diagnostics. So from here, I went to California and fell down a well called omics. And so omics is actually tools that give you information about all the parts of your body that we've been discussing. Genomics, transcriptomics, which is the transcription of your genomics, proteomics, which is the proteins that come from that translation of your genomics, and metabolomics. So what is your metabolism doing? And we're talking a lot more about metabolism these days, particularly in the context of COVID, because there's all this fatigue, there's other things that are happening with how you're struggling to live your life, basically. And that kind of comes down to, in part, your metabolomics. And so from there, I started thinking more about what's called pharmacogenetics and pharmacogenomics. And I worked with a company called the Gena Biosystems, and they were able to use mass spectrometry at the nuclear level, and we were able to look at specifically how your genes react to drug medication. So one of their most famous products is in at the VA, where they're looking at how to better use mental health drugs to target the right mental health patients. So if you've ever had this experience or worked or had someone in your life with the experience of trying to take medication for mental health effects, you kind of titrate on your own and you kind of try things until you see if they work. But with the advent of things like pharmacogenetics, you can actually look at how that pharmaceutical, that drug, is going to be acting upon specific parts of your genome based upon the data and evidence, and see that you could potentially have a better outcome from it. Now, the most useful thing that happened for me with this is there's a thing that actually measures the amount of your metabolic uptake of drugs in what's called CYP2D6. And some of us have more copies of it than others. And so I have way too many copies of this. So it was like, why can't I stay under anesthesia? Why am I no longer numb so very quickly from dental work? Like my dentist used to say, I have to give you 4, 10, 12 times as much anesthesia. I give you as much anesthesia as a linebacker, and you're a 140-pound, 5'6" woman. And it's because I'm metabolically upregulated. And I personally think this is actually one of the most important things that you need to know about yourself is your pharmacogenetic status. And I think it's really important to understand that in pediatrics, because somebody like me, if I were to take the wrong opioid, which is a time-release drug, I will over-metabolize that. And some of the things that you see with bad outcomes come from the fact that we just don't know someone's pharmacogenetic status. And I'd love to talk more about that. It's kind of a hill I will die on. Okay, so what I really think that one of the most important things I've kind of alluded to, data. Nothing that we were able to build would have been possible without really having good pools of data and being able to utilize that data. So we're talking about the uprise and advent of data scientists, bioinformaticians. I actually said that right. I've almost never been able to say that word right. And the use of the data that we're getting from the omics tools to understand how to make potentially predictive modeling. So, it's really about being able to use those data that we're able to access and look at them in new ways to understand outcomes better. And from that, we can then create a personalized approach based upon data about that particular genetic status. Or as this example here, a cardiac example in the 60s where we looked at different ways to understand how biomarkers and behaviors could actually affect outcomes of your heart. So I think that none of this really would be possible if we didn't have good data management. So where medicine really started to meet science fiction in my life was when I went to Abbott Laboratories, and we started using the genetic characteristics of patients, so using information about their genes to match them with a treatment that their genes will recognize or that can use a person's genes to improve their health. And one of the best examples of this is a drug based upon the genetics of cancer cells that's highly used across the board, but was first in my world in breast cancer. And this was a really interesting time in everybody's life. This was early aughts. And we started going into physicians and saying, chemotherapy of this, so this is a particular stratification of type of breast cancer when we first started using it. And what we saw was that these patients were not really getting well from the standard of care and chemotherapy, but if you added in a drug, and the first one we were talking about was Avastin, and from that they used Herceptin. If you added this additional drug on, then their survivability went way up. And we could prove that using FISH probes. So PathVision is one of the first IVT tests on market that was owned by Abbott Labs, and we were using FISH. And you can see here, like, normally you have 2 alleles, right? So you should have 2 copies of each of these genes, but if you're what's called upregulated, you got this whole bolus of these things, and this is potentially a candidate who would be responsive to Herceptin in her breast cancer. And so I was going around teaching this at hospitals to cytologists and going out and basically evangelizing the use of your genetic status to help give you better treatment and care from a specific type of medication. And this then became one of the first what's called companion diagnostics. So if you're familiar with how pharma companies and genomics companies are trying to work together now, you're looking at a potential biomarker, again, like genomics, and matching it to a drug outcome, and you want to build those 2 tools together so that you can identify the patients that will be the most, what we call responders to that drug in real time. So you attach a CDX, a companion diagnostic test, to a drug, and you use that diagnostic tool to understand the patients that should receive that drug. It's a very expensive process that fails a lot, but when it works, it's remarkable. So another thing that I was working on, this goes way back to Georgia Tech, targeted drug delivery. I mentioned it earlier, and I thought I would give another nod to my good friend, Dr. Prosnitz. We were building things that weren't the rule of our lab. We called ourselves the drug lab, even though we didn't make drugs. And we were the delivery lab, even though we didn't make pills or injections. You couldn't do those 2 technologies in Mark's lab. But what you could do is build microneedles. And microneedles are everywhere. And he actually, in this lab, in Mark's hand, has the first patents on microneedles. So it's accepted that all microneedle technologies that come down from this kind of originate here. At least that's Mark's argument. So we're gonna go with that one. But yeah, so one of the first ways that we designed that was in what was called lithography, to make metal tips, metal needles, and try to pass drug through, try to put things on the skin. And that actually wasn't the best concept because you're up against volumetric measurements and trying to get enough load into there. And so these are actually dissolvable needles so that you can put the patch into the skin, they dissolve and do a transfer rate. And this is to develop vaccine. So one of the things that we were working on in conjunction with the CDC was trying to create vaccine potential that doesn't have to be in the cold chain and that works specifically for you as a target on your skin and isn't an injectable. And so he's gotten a lot of interest in that work, and it's been really great to see that progress over 20 years. My work was in using, my friend used to say I used to play music to cells. So this is actually this is the famous image that he alluded to earlier in my publication. So I was using ultrasonic cavitation to expose transfer portals in the cell so that you could have therapeutics transfer in at a faster rate, or to make sure that the therapy only goes to those cells that are targeted by the cavitation for transfer. Because in the use of typical chemotherapies, it's given in through your vascularization. It goes all through your body and kills a lot of your fast-growing cells. You get all these side effects. Our hope, and there's still people who work on this to this day, is to take cavitation tools, expose the cells to the cavitation, and the transfer only happens in those cells of interest. And I was up against electroporation. There were a lot of theories about how this worked. I said it actually literally causes cavitation damage effects, and took very high-level microscopy images because I will take it to the mat to prove something. And so I became famous for these types of images and showing how sonoporation worked. So. And it's interesting because that actually became a big part of my career. The technology that I'm most famous for is my ability to do microscopy. So I worked for Leica Microsystems and helped launch super resolution ground state depletion technologies, where you break the diffraction limit and look at things very, very small that you can't see typically in light microscopes. So it was to take those sorts of images. So that was electron microscopy. So when you talk about looking at viruses and very, very small things in high resolution, you usually have to go to electron microscopy. And so this is using like a light-based tool, ground state depletion, to look at what's called super resolution, to try to get you into that same level of resolution without having to go to electron microscopy. So some explanation of that. One of the other things that I think is really interesting to talk about that's had a resurgence is what's called functional medicine. So 20 years ago, when I was working on these technologies and trying to show that you could improve cancer care by treating these cells directly, we were actually kind of making a bit of what's called a functional assay, where you take the cells or parts of the patient's body, expose it to the drug, and show that it would potentially make the patient better. But 20 years ago, we didn't have the compute power, and you're talking about taking cells from the body and trying to pretend that they're like the body. And so those outcomes were really not well predicted. But what's happened in the last year or two, really, there's been a complete resurgence in functional medicine. So I was working in this London-based company, and we were able to take cells from patients that had been diagnosed with specific types of cancer, and then we'd have them come back in and we'd take some more cells from them, or we would take part of their surgical resection, we would reconstitute it in a hydrogel, and then we would influx that hydrogel with the therapy, and then we would use computer vision and my favorite thing, confocal microscopy. So we would take three-dimensional microscope analysis after they've been exposed to drugs, do live dead staining, and then we would tell if that drug was going to be good for the patient. And that's in clinical trials right now in the UK. So, but it's been interesting to see that come back as a resurgence. A lot of things that we were trying to do 20 years ago, like gene therapy, other, this functional medicine that we really didn't have enough technology or enough understanding to do well back then, are definitely having a renaissance now. And then this is something that I'm pretty proud of. We hear a lot about machine learning, AI, ML, artificial intelligence. And this is one of the first products on market, based upon one of the first panels on market, that uses a machine learning algorithm to do predictions about matching patients to specific drugs. And this is the product that we built so that you can kind of reproducibly do that. So again, you need massive amounts of data, and you want to create a biomarker based around the data that you get from patients that were responding, so that you can understand more about how to find those patients who will respond in the future. And this is based in AI ML principles, and it's actually doing pretty well these days. So pretty proud of that. I think one of the things that people were most afraid of, and it is actually happening in real time now, is how I keep talking about those designer babies. So when I was at a company called Natera, we were working on what's called NIPT, non-invasive pregnancy testing. And the goal of that is to take a pregnant mom's blood sample and tell you about the status of the fetus without having to necessarily do the amnio yet. Is the pregnancy affected with Down syndrome, for example? The most common trisomy and the one that most people recognize, Down syndrome. So when I was at Abbott, we were actually doing karyotype staining, amnio sorts of staining, which are much more invasive. So this is this uses blood samples from a mom and is able to tell you about the status of the fetus quite early on, like early stages of the pregnancy. Now, again, the lot of moral and ethical quandaries that come from this kind of testing, especially in countries where you are time and rate limited on when you could potentially end an effective pregnancy. So we're trying to get these tools that can tell you in the first 9 weeks, in the first 10 weeks. But you're up against precision issues, and you're also against what's called reportable issues. This is a screen. So you should not be using this as a diagnostic tool to make a decision about a pregnancy. Unfortunately, it does get used. This is something that I think we should talk about. So using these kinds of technologies, we are understanding more about the pregnancy. But this is supposed to be a screen to tell you to take the next level of testing to really understand. You need to go to karyotyping, right? But again, time and rate limiting steps here make it very challenging about this process. The other thing that we're understanding more is looking in IVF at the embryo status. So you can kind of pick an embryo potentially that has characteristics or non-characteristics that you're interested in. And then carrier screening the parents themselves. And this is really important in specific populations like Ashkenazi Jewish populations, right? So again, we have the technologies now to kind of designer baby a bit. And in some places, they do make some claims, not in the United States, but I work globally. They do make some claims about telling you more about the potential baby that you're going to have. So, we're already kind of doing it now. And then this is a very well-known company. I really appreciate everything this CEO does. It's called In Silico Medicine. And they made a promise to each other a while back that they were going to work really hard to use AI ML tools to create new drugs and therapeutics, and they actually put one of the first on market. They focus in on some longevity, but longevity and oncology are very tied into each other, and they do a lot of different types of work. So they're literally kind of changing the future right now by using these tools and AI ML to help make more effective drug therapies from the outset. And I have, strong feelings about the use of AI tools. I personally think that it's good for my work because we're able to look at lots of data faster, get better resolution from the data. I think it's going to be useful to help people more. And so I pretty much embrace it in my work, and I'm very proud of the inroads that I've had so far to use it. And then getting back to how can we really, really change people? Well, there's this technology, again, mentioned called CRISPR. So we could potentially go in and edit and really design you to be somebody different. Are we anywhere near that? Not really. But there are some physicians overseas who claim to have affected fetuses, and the babies have been born with some use of CRISPR. Generally, CRISPR, though, is used for other things in diagnostics or in understanding, how to make better therapeutics. So there's cell therapies that are using some CRISPR. But in terms of actually affecting people, and we said this 20 years ago, gene therapy. We're going to go in and change your genes. So I don't think that's really an issue right now, but it does get back to the moral quandary of how do we really use these tools that I think about all the time. How are we really doing no harm? Are we really making sure that the drugs, therapies, AI, all of that is really doing the best for the patient? And that's a question that I ask myself every day that I work on it. One of the other things that I really like to say is that in one man's lifetime, if we look at Watson Crick and Rosalind Franklin and how they understood what DNA looks like, they were able to elucidate the structure. And then we were able to elucidate what it looks like in your body. And then we were able to target drugs and things to make you better. And now we're able to potentially change it. And so that's how fast this is changing. And this kind of walkthrough tells you just about my career in it since the 90s. And it's tremendous, the amount of changes that just I've seen. And I really appreciate the time you've taken to listen to me talk about one of my favorite topics. And if you have any questions, I am happy to talk about it.
Yes. Okay. Thank you. What are some hurdles that you're facing, or that the industry is facing, or sometimes in personalized medicine? Actually, I work in the industry. We make primarily microelectrode arrays, and also our subsidiary makes microscopes that analyze cells in wells,
Oh, wow.
multiple plates. So actually, this London-based company, I wonder what technology they're using.
We should talk more because I'm always interested in microscopes, and I consult for a lot of companies that have microscope tools. So we should talk more. Technical hurdles or business hurdles or social hurdles? I actually talk about it in terms of all things. So one of the interesting things about my career is I started as a technologist, got led into being a sales person, and then ended up becoming a business leader. It was not necessarily by design. I thought I'd still be sitting in a laboratory. I just had a knack for understanding a global view and demanding money from people. I actually am very good at negotiating things. But I actually see a lot of different hurdles. Now, my personal hurdles sometimes can be that I mentor a lot of early stage companies, and whenever you come in young, so there's a lot, there's so the rise of a young crew of CEOs. They have these great ideas, and they are founders of these great ideas, but there's not foundational knowledge of what it really takes to get all the way through to a therapeutic. So sometimes they're just up against their own challenges of understanding that it's going to take 10 years and that's going to take $500 million. So one of the biggest challenges is that getting anything on the market is extremely expensive, requires significant amount of data, lots of patient participation. There's a lot of things that come into play. We can start a diagnostic company tomorrow, and I could probably get some money for that. But to actually get all the way out to something that's going to affect patient care, that's best statistics 7 to 10 years and lots of money. So the first hurdle is everybody's got a great idea. How do you actually apply that idea in a way that makes sense? And then from a social standpoint, there's this constant thought, again, should we actually be doing it? So some people don't want to know if their pregnancy is affected. Some people don't want to know if they're potentially going to have Huntington's. And so there's different things about, do we really want to give diagnostics to people that don't want it? And then there's a lot of questions about the quality of the diagnostics. So FDA versus CMS, there's a lot of like alphabet soup in here, but when you want to do a reportable diagnostic to actually affect patient care, there's levels of evidence in there. And most things in the United States are under what's called an LDT, where you have to show practical outcomes and there's all these things that go from it. An IVD is a very high level of stringency that most companies cannot afford, and it would really block down on progress if everything had to be IVD. But there's a lot of talk right now at the federal level, the president's office, should we change that stringency requirement to make sure that the things that get on market are better than what's on the market right now. As all of you probably saw with like COVID, the emergency use authorization put lots and lots of tests on the market. How many of those were actually that good? So, that's one of the biggest challenges there as well. So right now, what are regulations going to say, and how do we get these on the market? And then from a personal level and from a technological level, it takes time. So like I said, there's a renaissance of gene therapy. There's a renaissance of these functional things. So even if you had a great idea, it might not be mature enough for prime time. We might not have the compute power. We might not be able to get enough data about it. You might not find enough people with that disease to get enough instances to even make the model work. When you're looking at rare trisomies of pregnancies and you want to get it from actual patients, you're talking about people at one of the worst instances of their life. So you're having to make like potentially artificial, things to measure. So there's just a lot of different factors that get involved. But I've seen many, many things get on market that do really great things for patients. And I've really only, in the time scales that I'm talking about, 20 years actually isn't that long considering how many things I've actually seen come to the market. Does that kind of answer? That's a big question with lots of thoughts, but I'm happy to talk with you more about your technology. Mm-hmm.
You mentioned about being able to test for trisomies with maternal blood. How does that work?
So what you do is you take a sample from the mom, and then you take it down to the plasma level, and you look for what's called cfDNA, cell-free DNA, and you can separate out by different SNPs things that are most likely the mom's DNA versus things that are most likely the baby's DNA. And then you can weight average that and look for a trisomy in what would be the fetal fraction of the DNA. Yeah, right. And so that's interesting too, because if you look at cell-free DNA companies, then they all start switching over to oncology companies. So Natera, I helped them become an oncology company because I came in when they were NIPT, but I had this big background in oncology, and a lot of cell-free DNA, there's a very common thread between a tumor and a baby in some ways, right? You're talking about DNA that isn't necessarily the host DNA, and so you're identifying unusual bits of genetic information. And so a lot of these companies that were NIPT companies also have cancer studies and cancer products too, because cell-free DNA is a very powerful thing to study. It's a weight of how many instances you have, so the mom's going to have more and the baby's going to have less, kind of thing, right? So you look at it from a position where it would be on the genome, and then you weight it out. I mean, there's other ways to look at it. So the original companies were looking at methylation status because you're upregulated in methylation when your body is changing, which is cancer, again, and pregnancy, and certain types of diseases. So part of the reason that they started thinking about this in terms of cancer is they were actually predicting some cancer in maternal instances. They were like, well, this looks really strange. And they're like, oh, that's actually cancer. So you can weight the different SNPs and find things that are probably not from your body, and that's how these liquid biopsies work. So Natera's product that I helped with is what's called a liquid biopsy. So we can look in your blood and understand about your tumor status and whether or not your tumor is going to recur, or more about like the specific genomics of your tumor to understand how to do better treatment on it. So liquid biopsy is a very big thing right now. There's many, many liquid biopsy companies, and so it's based upon the cell-free DNA concept. And where my use of it started out in the NIPT companies. Yeah.
You talked a lot about the evolution of biotechnology in Atlanta and Georgia.
Mm-hmm.
And so I was wondering what your thoughts are in sort of building infrastructure to how can we
So that's, I think, something I've thought about for a very long time. I stayed in Georgia longer than a lot of my classmates because I believed so much in the power of, Georgia Tech and Emory and Mercer and all of, Georgia State. There was this beautiful infrastructure in it. And we were hoping to create this corridor of new biotech startups. But part of what happened then was in '08, and the economy started failing. We all actually lost a lot of our investment. And one of the things that was trying to come in were large companies were trying to create a footprint in Georgia. Like, I believe Dendrion was one, Immucor was another. One of the reasons that San Francisco has such a good startup environment is in part because they have major players there as well. So you kind of need to have both. I think it would help if there was more infrastructure from either large pharmas or large diagnostic companies, because it is very hard to sustain an ecosystem that is a bunch of early stage startups that are hoping for their next VC round to keep their doors open. I can give a whole other talk about what it's like to keep hoping that you get that next fundraising round to try to not just pay your rent, but to make sure that your technology goes forward. So one of the instabilities in early stage companies is, can you keep getting enough evidence and can you keep getting enough data before you run out of funding? And it's really, really challenging to do. I think there's still a 90% failure rate on biotech startups. So I think, as much as I appreciate all of the biotech startups that are here in Georgia, and there's some really good ones that I know, to really get the attention and money and funding that it should be at, I think you're going to have to get a few more larger players in. But that's just personal opinion. It would be good, I think, too, with the way some things have changed with infrastructure here. One of the things that we were up against in part was some of the politics of trying to build biotech in Georgia. It was challenging. So I think that there's more openness now because in part the movie industry is here and the population is a bit more open-minded about technologies and tools and a lot more demanding, I think. I know with Hollywood here, there's going to be more interest in potentially longevity technologies. So a technology I didn't talk about that's got a lot of interesting players in it, and again, in silico is one of them, is longevity. How do we study your genes, the length of your telomere, things like that, to understand how much longer can we make you live? And that actually, I think, is interesting even to talk about women in menopause. There's some data now saying that if you could understand more about menopause and arresting menopause, maybe that would increase your health outcomes. So, it's a long answer to a short question, but right now I just think there needs to be bigger names, larger brands, or somebody here in Georgia with an early stage startup needs to get a great big check from an investor.
You mentioned about being able to detect the difference between cancer cells and regular cells just with the differences. And is the genetic differences enough? I thought it was more of an epigenetic change. It is like a significant measurable genetic change in the cell?
Well, it's looking at like the different pathways of the mutational burden as a whole. But yeah, so if you look at, again, the liquid biopsy technologies, you can measure out the different polymorphisms that are potentially from the tumor. But even if you look at if you're looking at like regular biopsies, the H&E staining, and then looking at the genetic component of it in IHC, and then to FISH, you're still looking at specific genes. And even with the HER2/neu, it's an upregulation of those particular genes. So you're still looking at how the genes are changing, or there's more of those genes of a particular type that shouldn't be there in the body. So it is still kind of a statistical measure, if that makes sense. But yeah, it's looking at those instances that make it different from you as a person, really. I'm not. If I can send you some papers if that would be helpful.
So you could use like the cell-free DNA in combination with other measures of the blood to kind of like as a combination of those.
Well, you could, but that's actually not what they're doing. But they are purely looking at the cell-free DNA. But if you guys want to have more information about it, it is based, again, on using that data analytics and the upregulation of these different changes in the polymorphism of SNPs.
So when gene therapy first was on the scene with the skin patients, there was a lot of enthusiasm.
Back in the day, we were using some kind of uncontrollable lentiviruses at times. But there is better ways to deliver the gene therapy. There is more understanding of what those genetics look like, and I think there's more stable constructs for doing that transfer. But it's not something that I do a great deal of these days, so, you know. And again, I'm on the business side more than the technical side, so that's kind of my high-level understanding at this stage about that condition. And if I am wrong, I will be happy to come back and correct myself.
You mentioned earlier about the microscopy of the cavitation, and in that picture there, didn't understand what we were looking at exactly in that slide.
Yeah, I'm happy to go back because I realized I was afraid I was running low on time. So this is an SEM image, scanning electron microscopy with a coating over the cell. So these are cell surface features in microvilli. And this is what normal cell substrate looks like, the normal surface features of the cell. Here you can see this kind of greeny area, and that's where we've basically kind of peeled away the normal cell area. And this is cytoskeletal infrastructure that actually makes it more porous. And so you can have an easier transfer of the drug or therapeutic into the cell. And this is like a 1 in a million shot kind of thing. If you look at the statistics on how it would be defined, I was purely lucky to even find this thing. But yeah, like this happens at higher levels of instance because of the nature of electron microscopy, you're looking at a cell per cell. And because of the nature of cavitation, you're looking at a variance of effects. And so it's like if you think about when a bomb goes off, everything closer could be decimated. Everything out here is probably more where you want it to be for good transfer, and everything out here is probably not really affected. And so if you look at, like, again, fluorescence microscopies and light microscopies and even flow cytometry was how we originally measured this, you can get more statistics and more instances of the cells of interest. But using EM techniques, I didn't know when I took this on because I was going to prove something. You're looking at each individual cell, and you're hoping to see a feature. And so I remember when I first saw this with Robert Epkerian, I was like, what is going on with that cell? That's so strange. What is that going on there? And I actually have a mirroring TEM images, which is the cross-section where it's a cut, and you can actually see, it's not of this cell, it's of a similar cell. And you actually see the outside of the cell membrane and a great big hole in the cell membrane itself. And so when I was looking at the TEM, I was like, that's a hole in the cell. So exactly. So I was very lucky where I did way too much of this microscopy because TEM is the same. You're looking at one cell. And since once I used these kind of ultra-structural measurements, then I was able to go back to confocal microscopy, which uses fluorescent dyes, and I could do like a wide scan. And so in fluorescent microscopy, you see these fluorescent rings with a hole in it. And so I have lots of those images. But these images are actually really special because they were on NIH website. They've been used in series, like movie series that talk about it because they're really such a good instance of looking at a cancer cell in EM. So, yeah. Yeah, isn't that cool? Like, I did this a long time ago, and I still get notes from people who are like, "Can I use these cell images?" Robert Epkerian was a professor at Emory who tragically died in 2006. And they named the microscopy, the EM Center, actually over him. And they named a forest in Armenia for him. And so I think every time somebody uses these images again, a little bit of him carries on. So, but thank you for that question. I think it's beautiful. It's beautiful work, and it was on the cover of the Boston Globe. It was actually highlighted in Georgia Tech. Like, it kind of made my career, and then made me realize I didn't want to do research anymore. I know, and I really feel like I need to go back and study some more about liquid biopsy because I'm just like, well, unfortunately, that was 4 companies ago, and I've done 18 techniques since. So, but I really appreciate those kinds of questions because it is important to understand how they're doing that, especially these days, because every time I turn around, there's yet another liquid biopsy company. And so I actually am at kind of the limit of my knowledge. They might be doing it a little bit different now. I helped launch Signatera, which is Natera's liquid biopsy, back in 2016. And that's a funny story. Like, I was going around the world doing tech transfer of NIPT, and the CEO was like, we really want to put this cancer product on the market. And we had a TED fellow, Dr. Jimmy Lin, who's one of the most famous oncologists in the world, actually, who was the chief science officer, and I was a commercialization expert, and they came to get me, and they're like, so you're the person who understands oncology and can help bring this to the market. So I got on a plane to London, and then the bridge burned down in Georgia So I'm in London, and that was when the bridge went down in Georgia. So we started talking about this to drug companies as an NIPT company, saying that we could help you understand more about disease in oncology, and it's really taken off. Like, that's actually kind of Natera's signature product now, Signatera, and it really does a lot of great things, not just for MRD now. Like, they've got really great research collaborations potentially to do what we call patient stratification. So as we keep talking about this in drugs, what we want to find is a person who's a responder to the therapeutics. You call it responder, and that's why my Genealis product is called Responder ID. ID does responders. And so they're actually using these tools like Signatera to try to understand more about those potential responders to a therapy and stratify patients before they go into clinical trials. So kind of the holy grail of improving your clinical endpoints is front-loading it by pre-identifying patients who would probably be the ones to respond so that you can get good clinical endpoints to get that out to the market.
And have some successes to demonstrate it.
Yeah, because one of the biggest challenges in getting drugs on the market is like making those clinical endpoints. A lot of drugs fail in phase 3, and that's actually one of the biggest challenges about those companion diagnostics. So you build a diagnostic tool that's going to help get this to the market by saying these are the right patients, and you're trying to get the drug to the market, and you still have to take it all the way through the process. And this is, again, millions and millions and millions of dollars are invested in this. And often drugs will fail in phase 3, and I've seen that take out, to Sam's question, these kinds of things are what takes out, like destroys these companies, is you make the wrong bet on a drug or you make the wrong bet on a tool. And so you get to this certain level of evidence that you need to get to the market, and it just doesn't work. So a lot of these tools now are being used to do what's called rescue drugs. So there's drugs with millions and millions of investment in it. They didn't quite make a clinical endpoint. And if we can look at what the data tell us about the patients in particular that did actually respond, or that the likelihood of those responses, and then again, preload a set of patients with a disease type that are gonna respond to that drug, maybe we can rescue that drug and get it to the patients.
Like a second phase 3?
Nope, starting again potentially to rescue it. Now, because again, the clinical trials have to be built in designation with endpoints. But there's drug candidates that had either good efficacy, but not enough. And so one of the thoughts is to take that drug that had great potential and maybe even use it in a different indication. Because again, if you're looking at what was actually practically working for that, or if you're thinking about how you can use that drug in different ways, you can use some of these tools on predictions and try to say, well, maybe this is a better patient population. So, it's become a really rising thing in the last, I want to say less than 10 years, but it's definitely become a rising thing in my experience, especially for companies that have large investment in these drugs. And so there's companies now that are incorporated around buying rights to these and trying to rescue them using these tools.
How do you balance the idea of like, oh, I know or I think based on the data that this drug will work really well for this very small subset of people versus a drug that will work kind of well for a large number of people, which
I really love this question because it gets back to one of the basic tenets of some pharma things about Blockbuster. So when you asked your question, what are challenges that you see? Rare drugs or rare disease does not get nearly the attention that it really needs. If you have a small subset of the population that's going to really, really benefit, but isn't going to be what's called total addressable market, it's not going to be a high TAM, that's going to be harder for them to get enough investment to make that actually come to fruition, because the stringency doesn't change. Whether you have a drug that's going to help 20 million people or 200,000 people, you still have to support it all the way through all these trials. And so drug companies are, again, the paradigm is they're commercial entities that want to make money. So it's hard to get something that isn't necessarily going to blockbuster. That's why there's lots and lots of diabetes treatments, right? Because that's a big blockbuster of a growing market. What I'm seeing happen now is that smaller companies are trying to step in. Like, there's a lot of small companies that are really just focused in on rare disease, And they've just made that what we call their thesis. So a thesis for the company and a thesis for an investment arm. And they're, positioning themselves as with the benefit of this will be so high to the people that need it that we're going to, make this our way of life. And so I think it's probably a better paradigm to do it that way than hope somebody like a major pharma company is going to take the time and attention to look at rare disease. Now, that being said, there's a very large company called Blueprint Medicine that actually took the time to look at a disease I struggle with. It's a mast cell disease, mastocytosis. And they actually, which is considered fairly rare, but is actually really probably not that rare. And they spent a lot of time making some drug options on that. So, but they're not Pfizer or Abbott or any of that. They're still relatively small, but they're a pretty large company. So, it really is one of those ethical questions, about the do no harm or do best effort versus the cost of drugs and capitalism and investors. Now, that being said, too, one of the things about treatment of patients, particularly in something like oncology, is that there's, these interesting biomarkers and CDXs, HER2, Herceptin, all that kind of stuff. But you don't have as many big blockbusters that really show great, strong effect in 60% of patient populations that often. You're more like 5%, 10%. And they will still prescribe that drug even if there's like a 5% chance of saving that patient's life. And one of the things that we use to motivate investors and to get money into this and to get to physicians who, one of the challenges is you're up against standard of care and liability. Right? So if you want to substantially change a physician's opinion, especially if it's going to cause a change to an outcome of a terminal patient or a patient that will be terminal, if you don't give them the right care, is you're really going to have to give them sufficient evidence to convince them to change that pathway, and that's not easy at all. Like one of the hardest things I'm up against, actually, is the liability of deviation from standard of care and the concerns, and I always understand this when I hear this from an oncologist, because they are making that decision about saving somebody's life. So technologically, are you really showing enough evidence to change what's an accepted standard of care? And so when we talk to the investors about how to make these tools better so that you can get more people, you can actually use a financial argument there. And so you can say, well, I can actually make this more than 5% of an expensive drug, 5% of treatment option of a very expensive drug. I can help these tools and maybe you'll hit 50% of people who need it, kind of thing. But also the drug companies, they're still getting paid for use. So even if it doesn't help 95% of patients, potentially they still want that prescribed because they're still getting that money in, right? So these are the constant things that you have to ask. Like, am I doing no harm on a 5% chance that I might help them with a potentially toxic medication, or am I doing no harm by telling them no when it might actually be something that can help them? And so that's, again, why these tools are so important and why it is very, very difficult in some ways to make people change practice, because if you're talking about fragile patients, if you're talking about terminal illness, you are harming somebody no matter what you do in some way. And that's actually, I think, one of the biggest take-homes of my entire career. Am I weighting the things I'm helping to get to the market do the best kinds of impact, or am I just making a capitalistic way of making people pay for more drugs, right? I think about that all the time.
Well, I think we'll be done. Thank you very much, Robyn. Yeah. So once again, thank you to Mutation for having us. We'll probably do this room again. Hopefully there'll be a little less crowd outside and a little easier to find the speakeasy, which is already a hidden room. But yeah, thank you very much for coming to this Science Junction. We really appreciate it. Our next event will be, for Science Junction, will be January 25th. We'll be at High Card Brewing in Tucker. And our executive director, Amy Sharma, will be talking about advocating for better beer laws in Georgia. Like how to advocate for anything. Environmental, science, beer laws is an interesting navigation of politics and the legislature here in Georgia. Being in Atlanta, we definitely have the capital, so there's a little bit more access. So that's going to be part of that talk. Hopefully then you'll be able to buy more than just a couple packs from your breweries. You might be able to buy a little bit more. So thank you again for coming out. We do this event for people like you, and we get support from people like you. If you would like to, scan this QR code and let us know what you thought. Really appreciate it, and thank you very much. Good night.